uh,
Hello, everyone. Hello.
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 Hello, ma'am.
Hello po, ma'am.
Alam, mapakiana lang ako. How did you, you listen, mapakiana lang. How did you submit your quiz?
Google form.
Through Google
Hitting the submit form.
Google
Pare
form.
ko kasi, hit santol, parang, tulong pala nag-hand-in. Nakikita lang din ha. Tulong nga nag-hand-in. Pero, waray ko nakikita ng attachment.
Ma'am,
I
through
can
Google
see
form
the, ano,
kasi po. Through Google form.
yun nga,
Dire po ba naka-attach?
dire, waray naka-attach.
Ano iba kasi
Transaction
nag-podcast
bank
na nala
naman.
tapos nakatuhirahan.
The site said
Chumat
that it's
Google
already
form, di ba
forwarded.
nag-send ka ko antas Google form.
Link, yes.
Kaman ng pag-answer din, submit malanam.
Opo, ma'am.
Waray ko kasi nakikita pa. Mango naman parang 500 in. Pero actually, what waray attachment sa kikitaon kung hawakuan. Kasi automatic ito niya makuan dapat. Automatic ito niya ma-check. Kikitaon ko lang ako ha. Anyway, so ano na lang nga how. So if I will ask you, anyway, instructions will be or announcements will be made later. So we'll start now with the lesson. Meiosis. Okay. Be ready with your notes and Be ready with your pen and notebook for note taking.
Ma'am,
You want me
excuse.
to? Yes.
Diri po yan naan pag-recap.
Yes. Oo, yan na. Anay, kay ko ang pa, anay. Mag-check pa magkadat attendance.
Sige po, ma'am. Sorry po.
Okay. Anay ha, check na anay ko attendance. When you say present, just say it aloud kay. Danay direct ko nababati. I'll start with section Mago. Apita, Zion. Just say it aloud, huh? If you answer, okay, very good. Araba, Mark, Vince. Mark, Vince. Araba?
Present.
Chad.
Present.
Chad.
Present.
John David Bantiles. Justin. Lance. Gabriel. Strobo. Go, Dick, Juno, Ernst, Von, Gabriel, Ardillo.
Present, ma'am.
Eugenio the fourth. Christian Andre. Marasigan. Alexi. Nicart. Jade. Ogale.
ma'am.
Carl Vincent. Yeshiel, Seso, San Emmanuel, Solayaw, Marie Vincent, Uy.
Present po, ma'am.
Okay. So, Bantiles, John David, not yet around. Justin, Bihar, Lance, Strobo, Dick, Go. Carl Vincent, Seso, Solayaw. Okay, we go to the girls. Aberia, Erica.
Present po, ma'am.
Lorraine, Abobo. Zoe, Afable.
Present.
Gail, Alde. Anna, Ambil.
Present.
Raven, Amoyo, Raven Rose. Amoyo, Raven. Ampong, Joyce.
Present po.
Joyce. Okay, Joyce is this Joyce?
Yes po.
Raven Rose, Amoyo. Raven Rose. Julia, Apura. Julia.
Present.
Okay,
Present.
very good. Nash, Astakaan. Nash. Angeline.
Present.
Lima. John Antoinette, Dator.
Present.
Krishna
Present po.
De Los Santos.
Present po, ma'am.
Jesslaine, Jolola.
Present po. Present po.
Aliana.
Present po, ma'am.
Jillian Gapate.
Present po.
Julia Guevara.
Present po.
Antoinette Labado.
Present.
Maxian Marasigan.
Present.
Maxian. So how do you pronounce your name? Maxian? Maxian.
Apo.
Okay. Debbie Menzon.
Present po, ma'am.
Isabella.
Present, ma'am.
Francesca Promeda.
Present po.
Jaslyn Puno. Jaslyn Faith Puno. Riz Sakmar.
Present po.
Angelique Suwal. Angel Cyril Monroy. Okay, I'll go back to the girls. Abobo Lorraine.
Tawag na tama.
Lorraine. Abobo Lorraine, present.
Reverse rama.
Ha? Abobo Lorraine. Kim. Not yet around. Zoe Afable. Zoe?
Present, ma'am.
Okay. Amoyo Raven. Amoyo. Nash, Astakaan. Nash. Suwal, Angelique. Monroy, Angel. Monroy. Okay. So we go to section Santol.
Ma'am,
Yes.
magta-travel daw po na iino.
Section Santol. Ay, klase nagtra-travel. One at a time. Klase nga ni klase. Pero kung matin ma-eskwela, face to face na. So, klase nga ni klase. Anyway, Joshua Abusejo. John Joseph Anyosa.
Present po.
John Edward Are. Mark Kenneth. Earl Catalo.
Present.
Earl Delantar. Earl. Labro Andre. Renz Lilia. Hans
Present.
Lomar. Lilia Renz, present.
Present.
Hans Lomar. Rosalejos. Hans Lomar. Raydel Samson.
Present.
John Ray Traboco. John Ray.
Present. Ma'am, present.
Okay, I'll go back to the boys. Joshua Abusejo. So absent. John Edward Are. Absent. Bulanadi Mart Kenneth. Earl Delantar. Andre Labro. Hans Lomar Rosalejos. Okay, girls, be ready. Princess Jane Abelia. Kera Adamas.
Present po.
Okay. Francis Alaya. Francine
Present po.
Alaya. Francine.
Present po.
Hannah Amos. Hannah. Denise Apura. Josel.
Present.
Who is this? Denise Apura, present?
Opo.
Okay. Josel Faye Banario. Andrea Joy Beato. Mishka Cepeda. Kiana Sesista.
Present po.
Kiana.
Present.
Jaze Ray Doldolea. Jaze Ray. Marian Dulfo.
Present, ma'am.
Cristile Factor. Fentes Denisa. Denisa May. Hyacinth.
Present.
Trisha Gapud. Trisha Nicole. Trisha. Angel Nadine.
Present po, ma'am.
Liana Gura.
Present.
Hazel Hilaria. Hazel, present. Shira Habuli.
Present po.
Shira. Charnet Latoha.
Present,
Charnet.
ma'am.
Giselle Mendoza. Giselle Mendoza. Jahara Montes.
Present po.
Isaiah Pagayanan.
Present po.
Alison May.
Present po.
Ann. Yeza Nicole. Yeza. Ninia Ramirez.
Present, ma'am.
Cherise Rijang. Cherise.
Present.
Grace Love Sabido. Grace Love. Urla Ella.
Present po.
Jazel
Present.
Padilla. Jazel
Present.
Padilla. Okay.
Present po.
Hannah Amos, are you there? Hannah. Giselle Andrea Beato. Beato Cepeda. Mishka Cepeda. Dolea Jazzy. Next, Factor. Fentes Denisa. Fentes. Gapud Tricia. Gapud Mendoza Giselle. Uno
Present
Yesa.
po Mendoza, ibang Letia Mike.
Okay, thank you. Puno, Yesa? Sabido Grace. Sabido, where are you? Okay. Astakaan is around. Okay. Nalaan na yan, may kulakikit taon. Italakit. Kasi taro sa kulinuwan. Gail Alde. Gail Alde. Okay, binati ako Gail. Oh, pika mo nga naging eskwela na nihirarayo ka mo. Naglaklase na nihirarayo. Ade, traveling to Bohol. Ba? Man. Okay, so again, be ready with your pen and paper for note taking. The discussion today is quite complicated. It is more complicated than mitosis, but I assure you, you will enjoy the discussion. Anyway, Hala, Sabido, and Mendoza. Now, today's discussion is about the stages of meiosis. But before we go to meiosis, actually this is just a continuation of, this is still cell division. Now this is another type of cell division, or a special type of cell division. Now, before we discuss the details of meiosis, we'll first hear from Aliana, who will give us a short recap of the discussion last time. Okay, Aliana, I am giving you the microphone.
Thank you po, ma'am.
Okay.
Good afternoon, ma'am. Good afternoon,
Good
classmates.
afternoon.
I give a recap of our lesson last October 15, 2021 on cell cycle and mitosis. Let us start with how cell divides by the process known as cell cycle. Cell cycle can basically be divided into two phases: the interphase, during which the cell prepares for division, and the mitotic or the end phase, during which the cell actually divides. The interphase is divided into three phases or stages: GAP1 or G1, S or synthesis, and GAP2 or G2 phase. And M phase is divided into two known as the mitosis and the cytokinesis. The cell spends its maximum time in interphase. During this phase, cell undergoes normal growth process, while also preparing for cell division, which takes in the cytoplasm. During the GAP1 or G1 phase, significant event happens. Cells grow in size, mRNA and proteins are synthesized, more organelles are made. In S or synthesis phase, significant events occur like DNA is copied or reflected. Each of the cell's chromosomes duplicates to become two identical sister chromatids attached at the centromere. In GAP2 or G2 phase, the cell continues its growth and get ready for cell division. A period of rapid cell growth takes place. mRNA and proteins are synthesized. More organelles are made and centrioles are reflected. Microtubules are also made. And the next phase is the M phase or the mitotic phase, which is the actual division phase. This follows after interphase. In this phase, cell divides to become two identical daughter cells. This phase is divided into four main stages: the prophase, metaphase, anaphase, and telophase. We can easily remember these four stages by the acronym pt. In prophase, chromosomes condense and become short, thick, and visible under microscope. Centrioles move to the opposite poles, mitotic spindle forms, and nucleus disintegrates. The next phase is the metaphase, which is divided into a prometaphase and actual metaphase. In prometaphase, nuclear membrane disintegrates and kinetochore microtubules are attached to the kinetochore of chromosome. In actual metaphase, the chromosomes are lying at the metaphase plate or equatorial plane. In the next phase, the anaphase. Here in this stage, spindle fibers shorten, centromeres divide, and sister chromatids separate and chromosomes move towards the opposite poles. In telophase stage, daughter chromosomes reach the opposite poles. Chromosomes unweave to become chromatin. Spindle fibers disappear, nucleoli reappear, and nuclear membrane reforms. The complete process of nuclear division, also known as mitosis. The next phase we have is cytokinesis. You should remember that in animal cell membrane of the parent cell pinches inward along the cell's equator until the two daughter cells form. And in plant cell, a cell plate forms along the equator of the parent cell, and a new cell membrane and cell wall form along each side of the cell plate. And lastly, we should also have to consider the important functions of mitosis for the replacement of damaged, old, or dead cells, development and growth of the organism, and regeneration of body parts in some organisms. That's all. Thank you very much.
Okay, thank you for a very comprehensive review from last meeting's discussion. Thank you for that, Aliana. Okay, so as what I said just a while ago, meiosis, the topic that we will discuss today is actually a continuation of what we discussed last time, mitosis. And as what I said just a while ago, meiosis is a special kind or a special type of cell division. Okay, so like mitosis, meiosis also follows certain stages. If mitosis involves the four stages, the prophase, the metaphase, the anaphase, and the telophase, which or the acronym PMAP, it is also observed in meiosis. Okay, are you all ready for meiosis? Yes or no?
Yes,
Ma'am. Yes, po, ma'am.
Po, ma'am.
Okay, be
Yes,
ready
ma'am.
with your pen and paper. So throughout the discussion of the topic, we'll be guided with these objectives. First is to describe its states of meiosis. Describe its states of meiosis. You will notice that each stage is different from the other stages. Another learning objective is to explain the significance of meiosis. So after the discussion, I hope you will be able to describe its stage of meiosis. I hope you will be able to explain the significance of meiosis. Okay? Okay, so we go to the stages now. Meiosis is divided into two major phases. One is Meiosis 1 and the other one is Meiosis 2. Again, meiosis is divided into two major phases or two major stages. One is Meiosis 1 and the other is Meiosis 2. Under Meiosis 1 are subdivisions or substages: Prophase 1, Anaphase 1, Metaphase 1, and Telophase 1. Okay? Meiosis 2 is also subdivided into prophase, anaphase, metaphase, and telophase. Now, since there are two major stages, tapos pareho lang itong era substages, you might as well specify which stage is that, or you are pertaining to, you're referring to. If you are referring to prophase of meiosis 1, you might as well write prophase 1. If you're referring to prophase of meiosis 2, you might as well write prophase 2. Duduha kasi itong major stages, tapos parehas itong era substages, meiosis 1 and meiosis 2. So pareho itong era substages, PMAT. Parehas PMAT. So to identify which is which, dapat bubutangan niyo in 1 or 2. So if you are referring to meiosis 1, you might as well write 1 for prophase, anaphase, metaphase. No. Balis ka din niya. Anay. I-correct ko lang. I-correct ko lang. This is prophase. Please make the necessary correction. This is prophase, metaphase, anaphase, and telophase. I'll make the necessary correction. Okay, malilito ka mo. So this is anaphase, metaphase. Well, this is anaphase. Okay, so we have it there. So prophase 1, metaphase 1, anaphase 1, telophase 1, if you are referring to meiosis 1. So these are the sub-stages. Okay. Now we go to prophase 1. Prophase 1. Now, prophase 1 is subdivided into leptotene. Please take this down. Prophase 1 is subdivided into leptotene, zygotene, pachytene, diplotene, and diakinesis. Again, prophase 1 is subdivided into leptotene, zygotene, pachytene, diplotene, and diakinesis. Again, these are substages or subdivisions. Now, since these are stages, please do not rearrange. Do not rearrange. It must always be in proper order. Again, leptotene, zygotene, pachytene, diplotene, and diakinesis. To give you an overview of the different stages of prophase 1, each stage is different from the rest of the stages. Magkaiba kada stage. Each stage is unique. Okay? Each stage is unique. Each stage is different from the other stages. Now, so makaka-notice ka mo, hala, meron yung sub-stages, 1, 2, 3, 4, 5. Of all the stages of meiosis, prophase 1 is the longest. This is the longest stage. So most of the time of meiosis is allotted to prophase 1. Dako itong iyan ako konsumo nga oras in prophase 1. Okay? Important processes are also found in prophase 1 or occur in prophase 1. Okay? Are you ready to go the details? Are you ready? Can we go to the details now?
Yes po.
Yes.
Okay.
Yes po, ma'am.
Again,
Yes,
again,
Yes po, ma'am.
just take down notes. Just take down notes. Okay, so we go to the first substage, the leptotene. Leptotene. Kung hag-Greek ya, in English kasi leptotene. Kung hag-Greek ya, this is leptonema. Leptonema, this is called as leptonema. Now, what happens or how do we describe the chromosomes in leptotene stage? Kasi itong division naman, itong cell division, it is more on the activities of the chromosome. The activities of the chromosomes. Now, what are the significant events under this period, under this stage? So what are the very important events or the significant events? Okay. During leptotene stage, please read 1, 2, 3, go.
Duplicated chromosomes.
Duplicated chromosomes
and
turns
long
into long strands that are more visible in the nucleus.
Okay. Okay. Now, the duplicated chromosomes condense into long, thin strands that are more visible in the nucleoplasm. Okay. So condensation. Please, hala, paki-turn off la. Paki-turn off la, please. Nadi-distract ako. Paki-turn off the microphone. Okay, thank you. So, this is the significant event going on or that occurs during the leptotene stage. So, these are the paired chromosomes. Paired chromosomes. Okay, paired chromosomes. Masarin ka, ma'am, kaya nga pair itong chromosomes. So, one comes from the father. One comes from the mother. So, one is a paternal chromosome. The other one is maternal chromosome. Itong akin kasi chromosomes, paniton hiya, 50% of it comes from the father. The other 50% comes from the mother. Ano? Okay, so next. Next is zygotene. Zygotene. Here, synapsis occurs. Synapsis occurs. So, mering nga ni synapsis, this is pairing of chromosomes, meaning that event that takes place under this stage is the chromosome will look for its pair. Magbibiling hiyahin iya padis. Magbibiling hiyan iya padis. As what I've said just a while ago, 50% of our chromosomes come from our father, and the other 50% come from our mother. How many chromosomes? Now, let me ask you, how many chromosomes do we have in the human cell? How many chromosomes are there in a human cell? Kera, anybody? How many chromosomes do you have in your cells? In each of your cells? Yes, Marie Vincent. Marie Vincent?
Accident la po ato, sir.
Okay, hala, Gail. Gail, how many? How many chromosomes do you have in your cells, in each of your cells?
1.
Gail? Kasi Gail is raising her hand. Okay, already. We go to Jillian. Jillian, Gapate? How many chromosomes are there in a human cell? 46. Very good. 46. Now, from this 46, 23 comes from the father, and the other 23 comes from, or the other 23 chromosomes come from your mother. So 50-50. Ano? 50-50. So iniya nga mga chromosomes, kada chromosome ay mga 23, let's say for example, ang 23 nga chromosomes, 23 nga chromosomes tikang hantatay, magbibiling hiniya kapareho. Okay? So synapsis, so that is what we mean by synapsis. Pairing of chromosomes. Pairing of chromosomes. Okay? Pairing of chromosomes. Magbibiling ito niya kapareho. So meron nga magbibiling ito niya kapareho, it iya bibilngon is homologous chromosomes. A homologous chromosome. Homologous. Magbibiling yan, so it's meron nga synapsis, magbibiling yan kapareho, kapareho. So meron nga kapareho, meaning they must be of the same size. They must be of the same size, and they must have the same traits. Kuha? Same size, same shape, same traits. Okay. So kung makabiling na hiya, parehasan kadako, parehasan shape, parehasan mga traits nga nakada, let's say for example, a trait for height, a trait for color, the color of the eyes, the shape of the nose. So parehas dapat iton aadli ito sa chromosome, itong mga traits nga adli sa chromosome dapat mahiaag-an liwa iton iya magiging pares. Once that chromosome will be able to find its pair, so we call that chromosome as homologous chromosomes. Okay? Homologous chromosomes. Similar but not identical. Parehas hira, pero they are not exactly the same. That is what we mean by homologous chromosomes. Again, in zygote, synapsis occurs. So pairing of chromosomes occur during this stage. Now, the paired chromosomes, which we call as homologous pair or homologous chromosomes, pair to form a tetrad. Tetrad from the word tetra. Tetra which means pera tetra, cherries. So meron nga tetra, peratia. So tetra pack. So tetra, peratia. Tetra. Yes, Jillian. Jillian? Jillian kasi una nga nakita nga nag-raise. Tetra means very good for homologous chromosomes pair to form a tetrad. Hala, ihapunta kon pera. So this is 1. This is 2, 3, and 4. Kuha. Meaning, may ada uupat nga chromatids. Siman nga ni kasi chromatid. I think you know what a chromatid is. They are exactly the same. Okay. Pero so meron nga ni homologous chromosome, the homologous chromosomes, the chromosomes are similar but they are not identical. Okay? Meaning, they are similar in terms of size, they are similar in terms of shape, they are similar in terms of traits, but they are not genetically the same. Mag-iba itong IRA genes. Please take this down. Again, please take this down. So, meron nga ni homologous chromosomes. Ano adto, Cherise? Homologous chromosomes. Ano adto, Cherise? Basta sa meron homologous chromosomes, how will you describe it? Cherise. Yes, Jocelyn?
Similar but not identical.
Similar but not identical. Similar in terms of size, shape, and traits, but they are not genetically the same. Their genes are different. Okay? Unlike the sister chromatids. Parehas ang genes, parehas tanan, exactly the same. Okay? So, asa itong pagkakaiba between sister chromatids and homologous chromosomes. Okay? So, in homologous chromosomes, maternal and paternal chromosomes pair. Maternal chromosomes and paternal chromosomes pair. Asya nga, may ada kita 23 pairs of chromosomes. Are you familiar with this? So human cells have 23 pairs of chromosomes. Meaning, if we add 23 plus 23, that equals 46 in all. Diba, Ana? Ano? Asya? 23. Bangin nagsasalatak math kay danay liwag akong mathematics. Asya. So 23 plus 23 equals 46. So the total number of chromosomes that we have in our cells is 46. So of this 46, 23 are maternal chromosomes and 23 are paternal chromosomes. So the maternal and the paternal chromosomes pair during zygote, a process we call as synapsis. Synapsis is actually a process. It is a process. Ano tiya nga proseso? Pairing of chromosomes. Ano ang nagpe-pair? The maternal and paternal chromosomes. Okay? Now, synapsis occurs, which forms the tetrad or bivalent. Tetrad or bivalent. Tetrad because uupat na ang iya chromatids. Bivalent kay ano? Duha nga chromosomes. Bi means two. So two chromosomes. Two chromosomes in a pair. Homologous chromosomes consisting of 4 chromatids. So that is what we mean by tetrad. Tetrad. So homologous chromosomes consisting of 4 chromatids. Okay, now you listen to this. Atin importante. When synapsis occurs, chromosomes lie on top of each other with a protein called Synaptonial Complex. Again, when synapsis occurs, chromosomes lie on top of each other with a protein called Synaptonial Complex. So, hindi nga Synaptonial Complex, asya ito nakakapagdukot haira. Okay? So, may itong protein aside from the kinetophore. Okay, this one. Oh, look at this. Look at this. Hindi nga white. This is a protein. This is a protein. Now, this protein, which we call as the synaptonial complex, joins together the homologous chromosomes. Okay, let me ask you, Hyacinth. In the illustration, how will you describe homologous chromosomes? or point out in the illustration the homologous chromosomes. Actually, na-point out na. So, ano pagkakaiba nila? Homologous chromosomes. Hyacinth.
Ma'am, similar po. Sira, pero diri ganoon ka identical.
Okay, look at the illustration. Look at the illustration. Okay, parehas. Parehas in size, parehas in shape. Now, when we say sister chromatids, exactly the same. Inihiya, parehas, hadi. Gets? Adihiya nga blue, parehas, hadi. So that is what we mean by sister chromatids. They are exactly the same. Similar in size, shape, and they are genetically identical. Okay, so we go to the next substage. The packaging. Pachinema. Okay, this is a very important, actually, pairing of chromosomes. Synapsis is very important also in cell division, because if synapsis will not occur, genetic exchange will not happen also. So in pachytene, genetic exchange between two non-sister chromatids, the paternal and maternal non-sister chromatids, chromatids from the maternal chromosome and chromatids from the paternal chromosome, cross over. They cross over. Okay? Ini kuntahiya, example ako ta ano, inihiya, chromosome from the father. Adi naman chromosome coming from your mother. Nagpade sira during synapsis. Now, since they are near each other, there is a big chance, there is a big possibility of genetic exchange. There is a possibility of crossing over, and when they cross over, they will exchange genes. Now, when chromosomes cross over, they copy each other's genes. So they exchange genetic material. Okay, kitai din hiha kitai. Inihiya nga pula nga hin green. And ira mga chromatids, and ira lower chromatid, the lower chromatid, the lower arm. Sigun ta din ang lower arm or longer arm. Nag-cross over. Pag cross over nira, nagibalyo hirahin genes. Nagikopya hirahin genes. Okay? Ano ang naging resulta? The green was able to copy the genes from the red. Okay, kita iha, nag-iba na hiya. The red in turn was able to copy the genes of the green. Kung ikaka dito, originally adihiya. Adi naman nga green adihiya, full green, adi naman hiya full red. After crossing over, this is what happened. Okay, dire na hiya Asia. Kuha. Dire na hiya Asia. Okay? Now, because of crossing over, that makes possible genetic exchange of materials or exchange of genetic materials. One thing good that happens is there is recombination, or it causes recombination of genetic information. It causes recombination of genetic information. kuha. Meaning, ang genes ang tatay nga chromosome, paternal chromosome, na transfer nga dihan maternal chromosome. Ang genes naman, han maternal chromosome, some of which were transferred into the paternal chromosome. Okay? Ano't mahinanabo when crossing over happens? Anybody? Ano't mahitatabo when crossing over happens?
Genetic exchange will happen.
The exchange genetic material.
Yes. When there is
Exchange
genetic
information.
exchange, yes. When there is genetic exchange, what will happen to the cells? Parehas la bahirahan original, magiging parehas bahirahan original. Cell.
Part of one
Magkakamay ada ba nila
chromosome
change?
another? Yes.
Magkakamay ada ba nila change?
Yes.
Yes or no?
Yes.
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 So they will now be different from the original cell. That an original kasi an mga pool, sigunta ini nga red paternal chromosomes, all red paternal chromosomes, all greens maternal chromosomes. After crossing over, after Packaging stage, where there is an exchange of genetic materials, wala, nag-iriba na ang mga chromosomes. Nag-iriba na hera. Meaning, after Packaging, it will now be a different cell. Maiiba na hiya. Okay? Maiiba na ang cell. Okay? Naiba na ang cell. Okay daw. Because of this, crossing over, that makes possible the exchange of genetic materials, variation of organisms happen. Asya nga, you are not exactly the same with your father. You are not exactly the same with your mother. You are slightly different. Signunta may ada kamo 5. May ada 5 nga siblings in the family. You will notice that you don't look exactly the same. All the 5 siblings do not look exactly the same. Parehas man ang gintikangan, parehas man ang nanay, parehas ang tatay. Now, the reason for that is because of crossing over, because of the exchange of genetic materials. Asya nga, may ada mo mga traits nga tikang kanin mo nanay, may ada mo mga traits nga tikang kanin mo tatay. Tapos ito nga mga traits danay diri na hibibiling di dahiton imo iba nga bugto. Okay? Okay. Now, the point or the part of the chromosome where genetic exchange, where genetic exchange happens is called as chiasma or chiasmata. Okay, look at this. Hinihiya. This point where chromosomes cross over is called as chiasma. This point here, hinihiya dida ngadapit. Luwag. Okay, so hinihiya dida ngadapit. This is the chiasma or chiasmata. Chiasma or chiasmata, they mean the same. So the part of the chromosomes where crossing over occurs or exchange of genetic exchange occurs is called as chiasma or chiasmata. Chiasma, singular, chiasmata plural. So chiasma ito niya, chiasma. So damo na nga ni chiasmata. Okay? So it is in the chiasma where alleles exchange genetic materials. Sabi nga ni alleles, magkaiba nga chromosome. Similar but not identical. That is what we mean by allele. Okay, so questions? Pakitin is responsible for the variation of organisms. Nasa master nga pa promotao kaya nga gi-riba kaya ano nga may white, may brown, may black. Pero mong tao, it is because of crossing over. Kaya ano nga nag-iiriba din at magburugto? Because of crossing over. Okay, so and it happens during Pakitin stage. Okay, so we go to the next. Diplotin. The diplotin stage. Here, the synaptonimal complex disintegrates. Ano pa daw ato ang synaptonimal complex? Antoinette Lavado? Ano pa daw ato ang synaptonimal complex? Ano ang iya gamit? Antoinette Lorraine. Hain ka na Antoinette Lorraine.
Ma'am, chromosomes.
Yes. Ano aton synaptonimal complex?
Ma'am, chromosomes lie on top of each other with a
No.
codes.
Dere, synaptonimal complex, ano tiya?
Ma'am, protein.
Again. Ano pa daw ato hala? Who will answer? Who will volunteer to answer? Yes, 3 participants are raising their hand. Maray na iba. Ana, sige. Ana. Synaptonimal complex, Ana.
Structure. A synaptonial complex is a protein structure in between homologous chromosomes.
So, synaptonimal complex is a protein that joins together the homologous chromosomes. Thank you for that, Anna. So, it is a protein that joins together the synaptonimal complex. Kay ito na homologous chromosomes, waray nga nito nakakapagpadukot hira. Segundo waray glue, magbubulag iton hira. So, para hira magdikit and crossing over will become possible, dapat may ada nira glue. May glue hira. Pero dire itong hiya glue ha. That is actually a protein. Okay, now during diplotin stage, this protein called the synaptonimal complex will disintegrate. Ma-weaken hiya. Ma-disintegrate. Kumbaga malulusaw. Kalusaw niya, what will happen? What do you think will happen? Erica. Nag-degrade ang synaptonimal complex that joins together the homologous chromosomes. So Erica Marie. So what do you think will happen to the chromosomes?
I think the chromosomes will separate.
Very good. And the answer is correct. They will separate. Magbubulag hira. Again, there is no forever. Nagdugot pero nagbulag-lag. Wow. Anyway, so the chromatids that were joined together through the synaptonimal complex will now separate. Okay, look at this. Okay. Dugot hira, meaning may dida synaptonimal complex and this chiasma. Later on, nag-disintegrate, nalusaw ang synaptonimal complex. So, nagbulag hira, napiritan pagbulag. Okay, pero pagbulag nira, dara-dara nira and genes that they copied. Okay, the genes that it copied from this red and the genes that it copied from the blue chromosome. Okay. Now, the synaptonimal complex will now degrade and the chromatids separate. So from this, di ba, they lie on top of each other, right? Okay, so magbubuglag na hira. Bringing along with them the genes that they copied. Okay, so these are the resulting daughter chromosomes. Okay, so meron nga bivalent, duha. So meron nga ni tetrad, o bivalent meaning duha nga chromosomes that are the same, that are similar but not identical. Okay, so meron nga ni tetrad, upat, meaning upat nga chromatids. 1, 2, 3, 4. That is what we mean by tetrad. Okay. So diplotin, the crossing over during packeting, tapos ma-degrade on synaptonial complex, and it will result to this. Nagbublag na hera. Okay. Next. Diakinesis. This is the last and final step of prophase 1. The last and final step of prophase 1. Here, the significant events that occur during prophase are observed in diakinesis. Ano ang mga significant events in the prophase of mitosis? The chromosomes condense, nucleolus and nuclear membrane disintegrate, spindle fibers begin to form, microtubule kinetochore attaches to the kinetochore of the centromere. Meaning, the significant events that take place in prophase are observed in diakinesis. Okay? Okay, questions? Questions on prophase 1. Feel free to ask. Antes kita maghinalaba, feel free to ask muna. I'm giving you 2 minutes for that. Questions? Wari pakiana. Bayan wari ka mapakaintindi. Sa wari na pakiana. Naiintindihan niyo? Again, yes?
Naman nag-discuss ka ang diakinesis ng putol-putol.
Putol-putol ako. Yan, okay na.
Okay.
Okay na.
Pwede po pakiulit.
Okay, sige. Dito in diakinesis, the significant events in prophase are observed in this stage. Ano ang mga significant events? The usual significant events in prophase. These are the significant events. Chromosomes condense. Nucleolus and nuclear membrane disintegrate. Malulusaw. Until such time nga mawawara hira. The spindle fibers begin to form, meaning centrioles will move to opposite poles. The centrioles will separate and move to opposite poles. As they separate, they will form the spindle fibers. Okay. So the spindle fibers will attach to the centromere. Ma-attach na hiya. All of this happen in diakinesis. Okay, balik kita ha. Balik kita. Haleptotin, okay pa akong microphone? Okay pa?
Yes, ma'am.
Okay. Okay, sige. Haleptotin, chromosomes condense. Zygotin, synapsis or homologous pairing occurs, which we call a synapsis. Okay. In pachytin, crossing over occurs, thereby exchanging genetic materials. In diplotin, synaptonial complex disintegrates and the chromosomes separate, or the chromatids separate. And as they separate, they bring along with them the genes that they copied. In diakinesis, the usual events in prophase are observed. And what are these significant events? Chromosomes condense further, kasi nag-condense na here during leptotin. So chromosomes condense further. The nucleolus and the nuclear membrane degrades or disintegrates. Spindle fibers form from the centrioles, and the microtubule kinetochore of the spindle fiber attaches to the kinetochore of the centromere. Okay, that is prophase 1. Medyo halaba, ano? Prophase 1 pala dito. Okay, so we go to the next. Metaphase 1. What happens during metaphase 1? Homologous pairs align along the equatorial plane or metaphase plane. Masa rin ka naman, Ma'am, di ba nagbulag na ithira? Nag-separate nga ni, pero are still together. Waray la nakakapagpa-attach haira, no attachment. Okay, so no attachment. Kay ano? Synaptonial complex is already degraded. Or synaptonial complex has already disintegrated. Nalusaw na. Nagbulag na nga ni, pero they are still together. Pair lagi hakira. Okay. Pair. Diras Joe, kung nagpapadispera din, yung mga ganyan. Tira mga drawing, dira mga asya, ikukorektan na lang. Okay, dapat din ito nila nagdudukot ha, dapat nagdudukot. Kasi nagbulag na ito nila during diplotin. Okay, now, there is phase, and that is homologous phase.
Yes,
Okay,
ma'am.
pa?
Yes, ma'am.
Yes, ma'am.
Next, we go to Anaphase One.